So I'll go ahead and get started. I'm Josh Bonkowsky, and I'll be talking today about new insights into cerebral ALD. Originally, I thought about titling the talk Keith Van Haren Part 2, but then I thought, well, that seemed a little bit disingenuous, so I had to come up with my own title. So let's see if I can do this. Nope. Sadly. Okay. So some disclosures, some grant funding, philanthropic support, clinical trial work, consulting, stock, and board of directors for a zebrafish company. So today's talk is kind of split into three parts. In the first, I'll give a overview to our approaches using big data or little fish, depending on the part of the talk that I'm talking. I'll talk about some of the challenges with understanding in cerebral ALD, and of course, how that limits then our ability for treatment and progress. And then some of the data regarding what we believe are some new insights into cerebral ALD. I apologize, I have two screens going, and so I just have to look at the left-hand one to move us forward. So I'll be talking first about big data and little fish, and you can see the big number 4 there, and that little tiny zebrafish. So what is big data for ALD? So really one of the challenges has been that prior to 2015, ALD was not separately listed in medical coding. The chief medical coding that was used is the ICD-9 codes, and then in 2015 it switched to ICD-10. And that was really helpful because then since 2015, we can uniquely know in medical records from data who has been diagnosed with ALD. There are still some limitations which make some of the data challenging. There's not separate codes for ALD or AMN or cerebral ALD. And then of course, sometimes the accuracy of the code can be questionable, and that requires kind of further validation work. It just makes things more challenging, but just the move in 2015 to a single code has been extremely helpful. So in the United States, there's several sources of big data for this kind of work that I'll be introducing. One is called FIZ, and I'll talk about that some more in a second. FIZ is only used, is only children's hospitals. And of course, much of the specialty care for kids is done at children's hospitals, but not all of it. And it's not every single children's hospital as well. There's also a database called KID, which uses a sampling method. And so it's representative of the entire United States, but it is limited a little bit because it is a sampling method. And so some of the granular detail is lacking. And finally, recently, some of the insurance claims databases are accessible, and these do have additional data, but they're quite expensive to use. And so they're just beginning to be available as a big data source. So the chief data set I'll be using today in some of our work is something called FIZ, or the Pediatric Health Information System. It's run by a system called the Children's Hospital Association, and was originally set up basically as a way to share cost data across different hospitals and try and get, for example, better deals on drugs or resources like gowns or masks or stuff like that. So it's a really large system. It has more than 50 children's hospitals. And then you can just see the numbers here, more than 2.2 million inpatients represented in the data, $90 billion of charges, 33 million different ICD codes. So just a very tremendous resource. So to kind of show you an example of the conundrum or problem we face and why we kind of launched in this research direction. This is a 13-year-old boy that I saw back in January who presented with, kind of classic cerebral ALD symptoms. He had subacute vision loss and difficulty in school. His exam showed a hyperpigmented skin and a significant loss of peripheral vision. And his brain MRI showed these, the bilateral symmetric T2 signal abnormalities with diffusion restriction and then rim enhancement. So these are the images, and you can see the T2 signal abnormality in the occipital lobes and then the rim of contrast enhancement. And this, of course, we were concerned, okay, this is cerebral ALD. And as we all know, there's significant issues with diagnosis and treatment of cerebral ALD. So we know that patients, even family members, even twin brothers with the same ABCD1 genotype can have distinct phenotypes, developing cerebral ALD or not developing cerebral ALD. There's never yet been a convincing second gene modifier identified, so we haven't found like a second gene that just determines like who goes on to get cerebral ALD and who doesn't. It does suggest that there's some sort of environmental or epigenetic risk determinant that we so far don't understand. And then, as you heard from Keith, Dr. Van Haren, there are shared features with other demyelinating disorders, in particular multiple sclerosis. Treatment cerebral ALD is it's not perfect, and it's really dependent on making a timely diagnosis, which sadly, as we know, can be really challenging and sometimes too late to help the child and the family. So this case I presented, which seemed like a classic cerebral ALD, ended up being multiple sclerosis. So after a lot of work, a lot of testing, he responded to treatment with IV steroids. He didn't have an ABCD1 mutation, didn't have very long chain fatty acid elevation, and has basically has tumor-affected multiple sclerosis. And so just showing, just from his presentation, the similarities between multiple sclerosis and cerebral ALD. Just as kind of an interesting side note and showing maybe how I did kind of a sloppy history, the reason his skin was dark is that he had been using a self-tanning lotion. I was like, oh, I guess I should have taken a better history about using self-tanning lotion. So as we heard from Keith, and as we know, the demyelination in cerebral ALD is characterized by a robust inflammatory response. And it's very similar, in fact, to the demyelination response we see in multiple sclerosis. So the hypothesis that has been explored by Dr. Van Haren and that we've also been thinking about is why is there's kind of this shared feature of demyelination with inflammatory components both in multiple sclerosis and cerebral ALD. In multiple sclerosis, there's this intriguing association with vitamin D status and the risk for multiple sclerosis, and it suggests that maybe low serum vitamin D could also be associated with the risk for cerebral ALD in demyelination. So in very nice work that Keith's group has done together with Patrick Alberg, Jerry Raymond, and Larry Steinman, they looked at low vitamin D levels and the risk for cerebral ALD. So this was a multi-year study with patients from Kennedy Krieger and from INSERM in Paris, France. And basically, they compared boys who went on to develop cerebral ALD versus those who did not, and looked at vitamin D levels in that time period leading up to the kind of endpoint of cerebral ALD, yes, no. And so in the boys who developed cerebral ALD, the vitamin D levels were significantly lower. And let's see if I can move hopefully this is not blocking your view, but the vitamin D levels were lower in boys who developed cerebral ALD compared to those who did not. And using several different statistical methods, something called a repeated measure analysis or logistic regression analysis, Keith's group showed that the odds ratio for developing cerebral ALD was lower in those who had higher vitamin D levels. And this was statistically significant with different types of analyses. So of course, this study did have some limitations. The sample size was relatively small, and it was difficult to do repeated vitamin D levels over the course of time. So in multiple sclerosis, as we heard from Keith, and as we know, there is a north-south gradient. And so the farther from the equator you are, the higher the risk of developing multiple sclerosis. And that's believed to be marked to correlate with vitamin D levels because sunlight converts the pre-form into the active form of vitamin D. So our question was, is there also a cerebral ALD north-south gradient? And so this is a map of the United States with the darker orange compared to lighter orange colors showing the ratio of cerebral ALD patients to the number of total ALD patients. And this is using that big FIS data set. So first we evaluated to see, was there any correlation between the risk of cerebral ALD and other factors? So for example, race or ethnicity, distance to the hospital, if you lived in an urban or rural area, the household income or the insurance type, and none of those factors correlated with the risk for cerebral ALD. So you could imagine that maybe being low income or living in a rural area somehow affected your risk for cerebral ALD. We did not find that effect. But we did find a correlation between the latitude and the risk for cerebral ALD. And so basically for every 2 degrees increase in northern latitude, there was an increased odds ratio of 1.17 that was statistically significant for an increased risk of developing cerebral ALD. And this is a graph of the data points and the logistic regression analysis for developing cerebral ALD. And so this is again from that big data set, the FIS data set, looking at the rates of cerebral ALD compared to the total number of ALD cases across degrees of latitude in the United States. So this data then is really helpful for kind of changing the way we think about ALD and cerebral ALD and what the risk factors are. But another major limitation in the field has been that there has not been a animal model for studying cerebral ALD pathophysiology. And this has been something that has been concerning to us as well, and something we've been trying to work on. And so the model we've been working with is called zebrafish or Danny Aurario, and this is a picture of the zebrafish. And it has key features that are really helpful. It's a small vertebrate animal, and because of its small size and relatively inexpensive cost, it's possible to large scale work with it that's just not feasible in other model systems. I would like to point out that it is a vertebrate just like you and me, and has the same genes that we do, including the ABCD1 gene. And so it's stuff that we find in zebrafish, we believe has direct applicability for understanding human conditions as well. So several years ago, now almost 3 years ago, we published our initial work on the zebrafish ALD model, where we made mutations in the ABCD1 gene. We showed that these zebrafish had elevations in very long chain fatty acids, just like in humans, and that there was a demyelination phenotype, also similar to what's seen in the AMN situation. One limitation with this initial work is that we didn't have a rapid demyelination phenotype seen in our initial model. So one thing that our postdoc in our lab, Quentin Ross, has been working on is to try and develop a cerebral ALD model. And his effort has been, is there some way to provoke a demyelination inflammatory response similar to what's seen in cerebral ALD? And he used a combination method of cuprazone, which is a copper chelating agent, together with a bacterial toxin called LPS. And so our simplest assay is where we look at the motor behavior of the animals. And in this, on your left here, the blue colored dots is a heat map of the zebrafish swimming, and more swimming is more blue. The wild type animals are on top, and the mutant animals after exposure are on the bottom. You can see there's much fewer blue areas, meaning that they're swimming much less. When we looked at the animal swimming, even wild type fish were affected by being exposed to cuprazone and LPS. But the important feature is that after exposure to cuprazone and LPS, there's a disproportionate worsening of motor swimming behavior in the mutant ALD animals. And so that's this group down here at the far right. And so they were worse, and they were significantly worse correlated with their genotype being ALD mutants. We also saw increased expression of inflammatory markers in the mutants. We looked at NOS2A and IL-1B, which are both inflammatory response genes. And we saw heightened responses following the LPS plus cuprazone treatment. Interestingly, it looks like there's a attempt at a compensatory response to the myelin genes. So different myelin transcripts following the LPS plus cuprazone treatment are disproportionately elevated in the mutant background. And so we looked at 3 different types of myelin genes, and they all had high elevations following the LPS cuprizone treatment, suggesting that there's a demyelination and then a compensatory response by the animals to try and recover. So if we want to tie back the work that we had looked at originally, looking at vitamin D levels and the risk for cerebral ALD, our prediction was that if we used a vitamin D receptor antagonist or something that would block vitamin D metabolism in the zebrafish, that we should see worsening of different features in the animal model. And so we used a vitamin D receptor antagonist called ZK159222, which we'll just call ZK for the rest of this talk. And so what we saw is that following treatment with this vitamin D antagonist, that there's significantly worse behavior in the mutants as compared to the wild type animals. So wild type animals are affected by this vitamin D receptor antagonist, but the mutant animals, even at relatively low doses, have a much worse response. So it suggests that not only do we have an ability to maybe model cerebral ALD in the mutant zebrafish, but that the vitamin D effect that Keith Van Haren has seen and that we've seen in our north-south gradient data might be something that we can also study now in the zebrafish model. So we have several experiments that we're going to follow up then to try and study this vitamin D effects. We have different agonists, which would presumably, we would predict, might improve the zebrafish behavior. We also have a different antagonist, which has a little cleaner biochemical mechanism called PS12192. And then our readouts are relatively straightforward ones, including motor behavior, the animals, RT-PCR for inflammatory markers, and then eventually myelin and very long chain fatty acids. So at this point, we have several kind of next steps in our work. We want to further characterize our cerebral ALD model, more completely understand the neuroinflammatory response, look at the effect on oligodendrocytes, on myelin, and then eventually also look to see like how prolonged is this effect seen. One of the challenges with working in zebrafish is that they are quite hardy animals and they have a fairly robust regenerative response. And so there is some recovery after even fairly severe insults, and so it could make our analyses a little bit more challenging. We're also embarking on doing transcriptomic analysis of both the LPS cuprizone model as well as the vitamin D responses to try and understand what are the key downstream pathophysiology mediating the cerebral ALD response, as well as where is vitamin D kind of intersecting that pathway. We'd really like to understand how is vitamin D impacting the disease, why is this happening? And we think there's some promising insights for something called the inflammasome, the NLRP3 inflammasome, which might be mediating some of these key inflammatory steps. So the conclusions I'd like to send us all home with today, first is that the clinical data as well as the distribution of cerebral ALD cases suggests similarities in mechanism to multiple sclerosis. This does provide some insights into understanding roles for vitamin D. And I think not only does it help us work, improve treatments for ALD, but it might also provide insights into multiple sclerosis. Also with our zebrafish model, we think we finally may have a model that helps us kind of understand cerebral ALD better. It's a unique tool for investigating pathophysiology, can help us understand both the inflammatory response as well as how vitamin D kind of intersects with the risk for cerebral ALD. I've had the good fortune to work with a lot of people over many years, and a lot of the work today was conducted by Quentin Ross, a postdoc in my lab, on the zebrafish cerebral ALD model. And thanks for your attention. I'm going to stop the screen sharing, but happy to see if there's any questions or anything in the chat box or in the comment section here. So, so we have a question from Artemio Ortega asking, are there any cures or treatments that can remyelinate the CNS and peripheral nerves and gene therapy for men with any type of ALD in the near future? And so I think that is really the key thing that we're all hoping to accomplish. I think between some of the chemical or compound screens that are being conducted, it's hard to know whether those just stop disease progression or whether they can help remyelinate. And then I think with some of the gene therapy or bone marrow transplant mediated gene therapy, that there is hope that they could also stop disease progression or even do remyelination. I think we all feel optimistic, but of course, 1 year optimism versus a 5-year optimism, I think we're all really uncertain at this point. Hopeful, but uncertain. And we have a question here to the panelists asking, are there any books I can get to better wrap my head around ALD? I will have to defer on that question. I'm not aware of any books. There's lots of online resources through ALD Connect, but I'm not aware of written books on the topic so much. We also have a question here about from David Moeller asking, do fish develop a pattern of CNS lesions similar to humans? That's a great question. We don't know yet. One of the challenges is that there's kind of two challenges. One challenge is that it's extremely difficult to do an MRI in zebrafish. And so to do an MRI on an adult zebrafish takes about 40 hours. And unfortunately, the zebrafish patient is dead at the time. So there's some limitations to that. For smaller zebrafish, the embryos, the imaging time takes about 3 days. So we don't know if the pattern is the same. The other problem that I mentioned briefly is that zebrafish have a regenerative response. And so we haven't looked yet to see whether the regenerative response is stronger than the demyelination effect. And so I don't know the answer to those questions, but those are important questions. There's some interesting questions from several folks here about vitamin D, and one question is, should people start taking vitamin D as a supplement or use a UV lamp? Another question is whether we should move closer to the equator. I think we'd all love to move to Hawaii maybe, but I think realistically, while the data is really encouraging, we've had examples in the past where we've had encouraging data and we've been just wrong. And so I think even though there's a lot of pressure to try and do things quickly, it's okay and more safe to wait in case we were just going off in the wrong direction for some reason. So I'd counsel waiting a little bit before we go too strongly with the hope for vitamin D as being a helpful thing. Here's another question, also similar, asking where they say they live in a rainy climate and asking whether they should get vitamin D levels from their pediatrician. I think that, again, at least with cerebral ALD, we're just not certain enough that we can offer, great clinical advice about whether to be checking levels, at least for ALD and for trying to do supplements. So folks, it's been a real honor to speak to you all today. And I hope I get the chance in 2021 and thereafter to meet many of you in person. And of course, if you're out in Salt Lake City, Utah, please come by, come visit the zebrafish. And have a great day and a great weekend here with ALD Connect.